interesting point. On the other hand, if we focus on some clinical study, we have several immunotherapy, and then including NKT adjuvant therapy. The NKT cell was invented the former president of Riken Center, so I'm somewhat involved in this one. And also, dendritic boosting is also another option. So, but they are usually developed under the very expertise of knowledge of experimentalists. So there are a few theoretical clarification and investigation on the effect of immunotherapy. So, and also I'm also interested in some ecology of tumor cells and immune cells. For example, IL-6 is known as a growth factor, as an inflammatory factor that initiates innate immunity against tumor. But in some later, so tumor can utilize IL-6 as a growth factor. So, and then macrophage can be utilized as a kind of a slave to produce a growth factor that can be useful for tumor. So there is a much complicated ecological interaction within the tumor microenvironment. So, and this is my final goal. So I've not talked about all of them. So today I just show that I focus on one of immunotherapy that is called adoptive T-cell therapy, and I analyze this population level model. So this is quite easy to understand, but I focus on showing the interpretation at the population level. And on the basis of this, finally I would like to incorporate these kind of piece of interesting topics. So I focus on ecological cellular modeling. This is described by simple mathematical model. So because I'm not sure whether the people are how much interested in immune system, but as far as I heard several lectures, people have already known about the immune system, so I'll skip this. But in short, immune system is a process of collective cellular behavior for defense. And this is a short summary of the cancer immunotherapy. Here I not included, I don't include cytokine therapy, but that is also important. But because I couldn't find a nice figure, so I skipped it. So here, basically, immunotherapy is classified into two types, using immune cell, or otherwise using monoclonal antibody. So one of famous monoclonal antibody therapy is targeting on the CD20. This is a typical marker of B cell, so that can be effective to lymphoma. This is a blood cancer cell, blood cancer B cell, and that can be shown to be effective. And on the other hand, there are several therapies that are classified into cell-based therapy, including adoptive T-cell therapy, dendritic cell therapy, or NKT cell adjuvant therapy. In most cases, we take immune cells from a patient and boost or activate ex vivo out of the cell, out of the body, and return it. That is why, because immune system recognizes, for example, virus or a virally infected cell, but tumor is initially given rise from normal tissue. So, and the T cell, for example, is one of major adaptive immunity, and they are educated within a timers not to react to self-antigen. The self-antigen is from the protein or peptide that from our tissue, but on the other hand, they can react to the outside of foreign protein. So, in that reason, in general, adaptive immunity against tumor is very ineffective because T cells do not react to self-antigen. That is one possible way. And also, immune system is finally activated under the process of self-antigen presentation or kind of co-stimulation, and under the presence of sufficient cytokine. So that is a very complicated process. So the boosting of T cell to become activated by by some clinical manipulation is therefore important to get effective immunotherapy. So today I focus on this. And I have already introduced the importance of ecological thinking. So now we have imaging analysis, and also we can detect, we can visualize, or we can observe the cellular behavior using fluorescent proteins or in vivo imaging. So, so now I focus on ecological model. That does not include spatial structure, but I show some simple ecological mathematical modeling that represent tumor and T cell interaction. So I don't, because I don't have enough time, and I will skip. But basically, so there are many studies in the field of mathematical systems or computational immunology, and so my current focus is cell population dynamics of tumor and T cells. So from here I show my current result. So this is a short introduction of adoptive T cell transfer therapy. So usually, T cell, existing T cell within in vivo are not sufficient to kill tumor cell. So clinically, we take tumor cell from a patient, and externally we add some cytokines that activate, for example, CD8 T cells that are also known as cytotoxic T lymphocyte, and then it returns it back to a patient. And then a part of this T cell can recognize tumor as enemy and start to attack and killing of tumor cells. So that is a basic idea. So to represent the interaction between tumor cells and effector CTLs, from here we call killer cell as CTL. So I describe the rule of population growth of tumor is described by logistic equation with intrinsic growth rate R and carrying capacity K. And this gamma represents the efficacy of killing of tumor cell by single cell CTL. And this is described by mass action law. And here we have two terms of the proliferation of CTL. Now I skip the very complicated process of becoming a memory T cells of CD8 T cells. But here I mix some effector T cells and also some memory T cells. And here alpha represents the rate, tumor-dependent proliferation rate. And this is also investigated as programmable proliferation term. This is tumor-independent proliferation rate of T cells. And here we have a constant inactivation rate. Here inactivation incorporates not only death of CTL by apoptosis, but also by inactivation. For example, PD1 and interaction, PD-L1 expressed on the tumor can become inactive of CD8 T cells. So we incorporate inactivation of such kind of process. And this basically, I put some nonlinear term with this. So you see that this is almost resemble as a classical Rotka-Portela system. But if you neglect this completely corresponds to classical Rotka-Portela system. So positive.
But then, yeah, I'm assuming it's positive, but that means that tumor in your model only stimulates the immune system.
In this case,
There is no suppression. I mean, a larger tumor suppression. That's what. That's why. That's not X. X only enters this one term. If alpha is positive, this means the tumor is only stimulated.
in this case, we assume that.
That's your assumption.
Yes, in this case, I assume, yes.
So you don't assume that if it gets larger, it will suppress.
And I don't assume any kind of, effect. So because I use simple, because we can provide a rigorous mathematical analysis on this system. And here is only the new part that I incorporate here. So I assume 3 types of functional form with respect to the programmed proliferation of CTLs. One is no feedback. So here just becomes a constant M. And in the second case, I consider negative feedback effect that represents a competition of ATC binding site at the dendritic cell, and via cross-presentation of tumor-derived antigen to CTL. And this term represents the autocline or paracline type proliferation of CTL. CTL can produce IL-2, and then IL-2 can become a stimulus for the proliferation of CTL. So that there is a kind of positive feedback proliferation. So these, on the basis of these biological consideration, I consider these 3 of function types. So first, let me explain the no feedback model here. So if we assume that the inactivation rate is greater than M, maximum proliferation rate, then here is a kind of negative death rate. So this corresponds to classical Rotka-Volterra model. And if we assume that M can become mu, then we have totally the following 4 possible equilibria. One is called naive equilibrium. In this case, we have no tumor cells and no tumor-specific CTLs. And I'm not sure whether the word is correct, but we call this anarchic equilibrium that represents the presence of tumor cells, but no CTL is activated against the tumor. So we have here, this is the carrying capacity. And we also have a Q equilibrium that represents no tumor cell under the presence of CTL. So we see that this is a kind of the establishment of memory cells against the tumor cells. So this is can be understood as a backsidation. And finally, we have persistent equilibrium in which both tumor and CTL exist. So in these 3 cases, we have already these 4 equilibria. And this is, excuse me. Yeah, and actually, yes, if we have a phase portrait here, and here, and here, and here, we have 4 equilibria. And this is a summary of existence and stability of each equilibrium. So, and this, I would like to derive some biological interpretation to this term and this term in the next slide. But basically, because this is a very simple planary differential equation, so we can show the global stability of each equilibrium. So let me explain the condition that switches stability from anarchic to persistent or persistent to cure. So actually, I first explained that M maximum proliferation of CTL is a kind of measure to know the transition, possible transition from no immunity to immunity, but tumor still exists, to finally tumor is eradicated. So we can, if M increases, steady state dynamics transit from this state to persistent to pure state. And here is the interpretation of the from equation. So we see that because delta does not make any effect on the stability and existence result for this system, but in the next another functional form, this can become important. And the delta star is considered as an adjusted density dependent inactivation rate. That is that is the addition of usual delta and something here. And this mu star is considered as an adjusted density independent inactivation rate. So this is determined by the difference between mu and alpha k. So assume that we have only tumor, then mu is the loss of CTL if we don't have any density dependent term. So if population of CTL is very small at the initial phase of therapy, so this is an intrinsic growth. Negative part is the intrinsic inactivation rate of CTL, and this corresponds to the maximum stimulation from tumor, because without CTL at the beginning of therapy, we could expect that tumor is in equilibrium. So we have K here. So alpha K is a stimulus from tumor. So it is determined as the sign of mu minus alpha K whether CTL can successfully proliferate to get a population number. So, and in the same way, we can interpret this as a re-emergence threshold for tumor cell. So here we see that if once therapy is successful, then we have no tumor and only memory CTL exists. So this is the concentration of memory cells. So gamma multiplied with the concentration of memory cell implies a pressure from existing CTL. And if tumor growth invades in body again, so this is the intrinsic growth rate of tumor. So the sign of them implies that whether tumor can successfully re-invade into the host body. So this implies that a patient has already been vaccinated, but again, if tumor appears, depending on the growth rate, intrinsic growth rate, it is determined whether tumor can successfully grow again or not. So we have, from a simple population model, we can get this kind of observation. So this is the scenario 1. And because I have to finish within 5 minutes, I will quickly explain scenario 2 and scenario 3. So basically, scenario 2 is qualitatively the same with the scenario 1. So we have here some negative density dependent effect in the proliferation term, but we have essentially the same set of equilibria. And also this is a summary of the result of existence and global stability of these equilibria. So one point that have changed from the previous scenario is here. The H is multiplied with mu star and mu. So nothing has not been changed. So H is a kind of the degree of control, some density dependent negative feedback. So this is just weighted. So essentially, qualitatively, we can get the same result. And also, we can, the transition of steady state dynamics is from no CTLs to CTL with tumor and eradication of tumor. And I have also investigated whether this term is from the composition of APC binding site at the dendritic cell, but I skipped the detail of explanation. But we can also have some possibility that this kind of competition can be advantageous for CTLs. So under this condition, but I will skip to explain in detail. And finally, this is the third case. I incorporated the effect of positive feedback model. And this term is derived from another subsystem. That is the interaction between cytokines and the CTL. The cytokine is produced from CTL, and the cytokine increases the production rate of CTL. So there is some kind of positive feedback. This is also known as autocrine or paracrine interaction. In that case, by using quasi-steady state approximation, we can reduce the equation for cytokine, and then we can get a single equation. So finally, we get this type of positive feedback function. In this case, we have the following 4 types of equilibria. They are unchanged, but this QI equilibrium and the persistent equilibrium can become saddle type equilibrium and node type equilibrium. So this can be easily understood because if positive feedback fails, it shrink. So this is a summary of existence and stability condition. In this case, we don't have no more global stability result because we can essentially have some saddle node bifurcation and bistability of 2 equilibrium. So, but still we see that kind of same kind of interpretation is possible in terms of the switching or transition from pure state to persistent or something. So in this case, we have 2 possibilities depending on the sign of mu star. If it is less than 0, it's simple. We have from persistent to pure. But on the other hand, if mu star is positive, then we have this kind of scenario. So it depends on the choice of initial condition. So first we start anergic. So no CTL response, but CTL response can be activated if we choose a sufficiently large number of initial CTL, and then it can become cure. But we still have the possibility of the failure of treatment. And so then tumor can exploit this mechanism as an escape from CTL. So for example, if this is a scenario that I copy the same scenario here, so this is described in terms of parameter m. So here, pure state at the present here, no tumor and only memory CTL exists. But if I decrease m from, then we see persistent, and finally we see anergic, so only tumor can exist. And this is a scenario if we focus on m, and we can show the same scenario if I change parameter k. This is from the parameter from tumor. So if tumor shrinks and antigenic stimulation to CTL becomes low, then there is a possibility of escape from immune system. But this is too oversimplified, so I don't say that this can really occur. But there is a possibility that if immune reaction is mediated by positive feedback interaction, so we may have a possibility of the escape of tumor from immune response. So this is the end of my talk. Thank you very much.